Our skeleton comprises over 200 bones, and not only serves as a support structure, but is also the body's largest calcium store and the site of blood cell production. Bones can be distinguished by their shape. For example, the cube-shaped carpal bones are short bones. There are also flat types, which include the shoulder blades. Another group is the long or tubular bones. In addition, there are sesamoid bones, which are usually located near joints and embedded in tendons. Irregular forms, such as the vertebrae. And not to forget the pneumatic bones, which are characterized by air-filled cavities. Let's take a look at the macroscopic structure of the femur. It consists of a bone shaft, also known as the diaphysis, and the epiphyses located at the ends. The metaphyses lie between them. Almost the entire bone is covered on the outside by a special membrane called the periosteum. Only the joint surfaces are excluded. The periosteum is well supplied with blood and rich in nerve fibers. The joint surfaces, in turn, are covered with smooth cartilage, which, together with the lubricating synovial fluid, significantly reduces friction. This fluid also supplies nutrients to the cells in the cartilage tissue. The bone is relatively light due to its special structure. It has a dense outer layer, the so-called substantia compacta. Inside, however, it has a spongy structure. This design saves bone material while still providing stability. In the diaphysis, we find the medullary cavity. In adults, this cavity is filled with yellow bone marrow. Unlike red bone marrow, it is no longer capable of producing blood cells. Bone is a mineralized connective tissue and shows a complex architecture at the microscopic level. In the compact bone, distinct structural elements can be identified. They are aligned along the longitudinal axis of the bone and are known as osteons. Osteons are about 2.5 millimeters long and contain a small central canal that houses both blood vessels and nerve fibers. Surrounding the Haversian canal are multiple layers of bone substance. Between these bone lamellae, non-dividing cells, the osteocytes are embedded. Osteocytes remain connected with each other through their long cell processes. Those closer to the center establish connections with the Haversian canal. The Haversian canals themselves are interconnected via Volkmann's canals. The spongy bone is composed of trabeculae, which also display lamellar organization. However, in this case, the osteocytes are nourished by blood vessels located on the outer surfaces. The cavities of the spongiosa are filled with bone marrow. In addition to the periosteum, there is also the endosteum, the bone membrane that lines the inner surface, as well as the Volkmann's and Haversian canals. Contrary to popular belief, bones are not static structures, but undergo remodeling processes throughout life. This is a basic prerequisite for their stability. Bone substance in need of repair is continuously replaced. In addition, like any tissue, bone adapts to changing conditions. More bone tissue forms in areas that are subjected to greater stress. Osteoblasts and osteoclasts are responsible for these remodeling processes. Osteoclasts break down tissue, while osteoblasts build new bone substance. I have created a separate video on my channel about this. Don't want to miss any of my videos? Then don't forget to subscribe. And if you liked the video, I'd really appreciate a like. It would really help me out.